An animal or plant starts its life as a single cell, the fertilized egg or zygote. During development, this cell divides repeatedly to produce many different kinds of cells, arranged in a final pattern that is both spectacularly complex and also very precise. The goal of developmental biology is to understand the cellular and the molecular mechanisms that direct this amazing transformation. Now, the embryonic development of organisms has fascinated people for a long time, even ancient philosophers like Aristotle, who lived about 300 years before Christ. The reason for this fascination with development is understandable. After all, an embryo builds itself, it's a machine that has to function before it is fully assembled. Development is the process of progressive change via which an organism goes from genotype to phenotype. Although the study of animal development has traditionally been called embryology, which is the phase of an organism that exists between fertilization and birth, development does not stop at birth or even adulthood because most organisms are always developing in some way. Developmental biology is not only concerned with the study of embryonic development, which happens before birth, but also with other developmental processes like regeneration, the replenishing of tissues from stem cells, metamorphosis, aging, and even the stages of cancer development, which are all things that happen after birth. So, developmental biology includes embryology in addition to these other processes. Although the main focus of this class will be embryonic development, we will examine some aspects of post-embryonic development later in the semester. The process of development results in the creation of different cell types that are organized in a specific way to produce a new organism. So it obviously ensures the continuity of life. And the field of developmental biology seeks to answer two fundamental questions. Specifically, how does the zygote give rise to the adult body? And then how does this new organism eventually produce another body? And within these questions, there are more specific questions that developmental biologists seek to answer. Among these is the question of differentiation. So how does a cell become specialized? How do cells differentiate? Now, all cells in an organism, except for few, like germ cells, have the same genome. So how is it that these same genetic instructions produce all of these different cell types? How does an organism go from a single cell, the zygote, to having many different types of cells? With this question, we also have to take into account that there are cell types that are only present during embryonic stages and undergo apoptosis after their functions during embryogenesis have been accomplished. As we'll learn throughout the semester, at each stage in its development, the cell encounters a limited set of options and its fate is influenced by other cells, especially its close neighbors. The next question is, how do differentiated cells organize themselves into complex structures, which is a process that is known as morphogenesis. Now, even though there is a lot of cell migration, cell division, and differentiation that occurs during development, in all these movements, these structures that are produced are highly ordered and they're predictable in the sense that certain structures in the body are localized to specific regions. For example, our brains are always inside of our skulls. They don't appear in some people in their abdominal cavities. Now, this creation of ordered form, this morphogenesis, involves the coordination of cell growth, cell migration, and even cell death. And morphogenesis is critical for the creation of complex tissues and organs like the brain. Developmental biologists are also interested in the regulation of growth. More specifically, how is it that cells know when to stop dividing? This is important in order to allow for our tissues and organs to have the correct dimensions. For example, both of our legs have to be about the same length in order for us to walk properly. Now, these figures show an example of what can happen when pathways that are important for regulating cell division are altered. In Drosophila, which is the organism pictured here, the loss of a pathway, which is known as the hippo pathway, very appropriately, results in a hippopotamus-like phenotype. So when this pathway is lost, specifically in the cells that give rise to the wing structures, the cells divide more frequently and they also die less. So you end up with structures that are much larger than the wild-type counterparts. These results suggest that there are key signaling pathways that are responsible for controlling cell growth. In addition to being concerned with the processes that take place during embryonic development, developmental biology is also concerned with the process of fertilization and the formation of germ cells, more specifically, the special characteristics that allow the sperm and egg to form a new organism once they combine. Well, besides the fact that each of these germ cells has, as you know, half the number of chromosomes and unique combination of alleles because of the recombination that happens during meiosis, as you'll learn later in the semester, not all paternal and maternal alleles are equivalently expressed. So we inherit some alleles from our mom that will never be expressed, and some alleles from our dad that will never be expressed. But I'm going to leave it at that for now because we're going to talk about this in more detail in a couple of weeks, and this is actually one of my favorite topics. As developmental biologists, we're also fascinated with the process of regeneration.